Today we're going to be talking about innate immunity, which is a defense that is active immediately upon infection and is the same whether or not the pathogen has been encountered previously. Innate immunity includes an outer covering, such as a skin or shell, that provides a significant barrier to entry by microbes.
Basophils are also granulocytes that attack multicellular parasites. Basophils release histamine, much like mast cells. The use of histamine makes basophils and mast cells key players in mounting an allergic response. Granulocytes are a category of white blood cells characterized by presence of granulocytes in their cytoplasm.
Next, we have a monocyte. A monocyte consists of a nucleus, lysosomes, and the cytoplasm. The purpose of a monocyte is to carry anti-tumor, antiviral, antibacterial, and antiparasitic immunity, and they are involved in regulation of hematopoiesis.
Next, we're going to talk about macrophages. Macrophages are very efficient phagocytic cells that leave the circulatory system by moving across the walls of the capillary vessels. The ability to roam outside the circulatory system is very important because it allows macrophages to hunt pathogens with less limits. Macrophages can also release cytokines in order to signal and recruit other cells to an area with pathogens. Cytokines are a large group of proteins, peptides, or glycoproteins that are secreted by specific cells of the immune system.
Neutrophils are phagocytic cells that are also classified as granulocytes because they contain granules in their cytoplasm. These granules are very toxic to bacteria and fungi and cause them to stop proliferating or die on contact.
Dendritic cells are antigen-presenting cells that are located in tissues and can contact external environments through the skin, the inner mucosal lining of the nose, lungs, stomach, and the intestines. Since dendritic cells are located in tissues that are common points for initial infection, they can identify threats and act as messengers for the rest of the immune system by antigen presentation. Dendritic cells also act as a bridge between the innate immune system and the adaptive immune system.
Mast cells are found in mucous membranes and connective tissues and are important for wound healing and defense against pathogens via the inflammatory response. When mast cells are activated, they release cytokines and granules that contain chemical molecules to create an inflammatory cascade. Mediators, such as histamine, cause blood vessels to dilate, increasing blood flow and cell trafficking to the area of infection. The cytokines released during this process act as a messenger service, alerting other immune cells, like neutrophils and macrophages, to make their way to the area of infection or to be on alert for circulating threats. Histamines, then, are chemical produced and stored within the body. It is a part of our immune response and is released during an allergenic reaction.
Eosinophils are granulocytes that target multicellular parasites. Eosinophils secrete a range of highly toxic proteins and free radicals that kill bacteria and parasites. The use of toxic proteins and free radicals also causes tissue damage during allergic reactions, so activation and toxin release by eosinophils is highly regulated to prevent any unnecessary tissue damage. While eosinophils only make up 1 to 6% of the white blood cells, they are found in many locations, including the thymus, lower gastrointestinal tract, ovaries, uterus, spleen, and the lymph nodes. Next, we're going to talk about toll-like receptors. Toll-like receptors bind to fragments of molecules that are characteristic of a set of pathogens. So each mammalian toll-like receptor recognizes a molecular pattern that are characteristic of a group of pathogens. Lipopolysaccharide, flagellin, CpG, DNA, which is just DNA containing unmethylated CG sequences, and double-stranded RNA are all found in bacteria, fungi, or viruses, but not in animal cells. Together with other recognition and response factors, TLR proteins trigger internal innate immune defenses.
Natural killer cells, also known as NK cells, do not attack pathogens directly. Instead, natural killer cells destroy infected host cells in order to stop the spread of infection. Infected or compromised host cells can signal natural kill cells for destruction through the expression of specific receptors and antigen presentation.
Now we're going to talk about the complement system. The complement system is also known as the complement cascade. It is a mechanism that complements other aspects of the immune response. Typically, the complement system acts as a part of the innate immune system, but it can work with the adaptive immune system if necessary. The complement system is made of a variety of proteins when inactive circulate in the blood. When activated, these proteins come together to initiate the complement cascade, which starts the following steps. 1. Opsonization. Opsonization is a process in which foreign particles are marked for phagocytosis. 2. Chemotaxis. Chemotaxis is the attraction and movement of macrophages to a chemical signal. 3. Cell lysis. Cell lysis is the breakdown or destruction of the membrane of a cell. 4. Agglutination. Agglutination uses antibodies to cluster and bind pathogens together, much like a cowboy rounds up his cattle. Okay, so the steps of the complement cascade facilitate the search for and the removal of antigens by placing them in large clumps, making it easier for other aspects of the immune system to do their jobs. Now off to the lysozyme. A lysozyme is an enzyme that breaks down bacterial cell walls and further protects the digestive system. It's seen in egg whites and in the nose. Thanks for listening. Now that's the innate immune system.